So all the programs which were completed earlier were of high quality, and most of them are reputed scientists from different national institutes. And today also, it is very fortunate that Dr. Srinivas Reddy has accepted to be the guest speaker for today's program. On behalf of Honorable Vice Chancellor, Dr. YSR Horticulture University, all university officers, and this program course directors, organizers of this program, I invite you, Dr. Srinivas Reddy garu, for this program. May I now request Dr. Jayaprada to kindly say a few words about today's guest speaker. Kindly introduce Dr. Srinivas Reddy to the participants of today's program.
Thank you, sir. It's my pleasure to welcome all the participants join for today's 24th session of the Citrus Graduate Readiness Program for Agriculture and Horticulture UG students. As the sir informed, this is especially for the benefit of ELP students who are in the final year of their undergraduate course. It is an honor to me to welcome Dr. T. Janakiram Garu, our Honorable Vice Chancellor, Dr. YSR HU, Dr. A.S. Padmavatamma Garu, Dean of Horticulture, Dr. YSR HU, who is the mainstream of inspiration and constant support for all the activities of our college. I welcome all the respected university officers, Dr. D. Srinivas Reddy Garu, Senior Scientist Entomology from Citrus Research Station, Tirupati, today's guest speaker, and associate deans from all the colleges of the university, and Dr. K.T. Venkatramana sir, Principal Scientist and Head, HRS Anantarajpeta, Dr. R. Nagaraju Garu, Principal Scientist and Head, Citrus Research Station, Tirupati, Dr. C. Madhumati Garu, Principal Scientist and Head, Citrus Research Station, Petlur, the course coordinators, organizing committee members, and all the faculty members of Dr. YSR HU, and our beloved students, and all the other participants joined for today's program. I take this opportunity to introduce our eminent speaker of today's session, Dr. D. Srinivas Reddy Garu, the Senior Scientist Entomology, who is well known for his outstanding work in all the horticultural crops, and also for his interaction with the farmers of this region. He is a popular scientist in this region. With your permission, sir, I will read out the biodata of today's guest speaker. Dr. D. Srinivas Reddy Garu has completed his UG course at UAS Dharwad, and to know his excellency in his academics, I would like to inform you that he got the Merit Scholarship Award by UAS College of Agriculture for his highest OGPA in the UG program. And he completed his MSc and PhD programs at IARA New Delhi, and he got in the entrance exam, he secured first rank in the entomology discipline at MSc level entrance exam, and he secured second rank in the discipline of entomology for the PhD entrance exam at the All India level. Regarding his professional experience, he has got a vast experience in both private as well as university side experience. He worked for the private sector before he joined here in the university. In the private institutions like Bayer India Limited, DuPont India, and Nagarjuna Agri-Chemicals Limited in research and development activities. In the university, he has the experience of 12 years, more than 12 years. He worked as, he joined as a scientist entomology at HRS Anantarajpeta, and worked on pest management activities in the crops like mango, banana, betel vine, guava, and melons. He developed the pest management programs and schedules in mango hoppers and twist weevil, as well as fruit flies, stone weevil, and midges. And he developed the IPM module schedule for the melons while he was working with the HRS Anantarajpeta. And he worked on mites management in betel vine and cucumbers. He established a biocontrol laboratory and laid a foundation for mass production of various bioagents and supplied them to the needy farmers in this region. In the academic line, Sir has taught the courses at UG and PG level at College of Horticulture, Anantarajpeta, and he guided more than 15 students at PG level. And he has dealt with all the courses that are related to pest management and entomology courses. And along with the research and academic activities, he has a vast experience in the extension activities also. He participated and some of them he conducted the training and workshops, more than 20 at various levels. And he attended the symposium and conferences, more than 12 at national and international level also. Along with the research activities, all his works were published in reputed scientific journals. More than 48 publications he has in the reputed journals. And he presented abstracts, more than 20 at the national and international conferences. And he published the books, more than 4, I think some are in the line. The textbooks which are very much useful for the competitive exams at the horticultural students, like the titles of the books I read, a textbook of applied entomology, which is published in 2003 and revised in 2014. A guide to ICR. I request all of you to please mute. A guide to ICR NTS exam, a concise dictionary of entomology, and a textbook titled as Pesticides, which is recently published in 2016. Along with these books, he has written book chapters regarding the insect pests and diseases of papaya and their management, and he published many technical bulletins. And along with the intention to help the farmers also, he published some of the books in Telugu language also to help the farmers. Many pamphlets and leaflets were published in his work to help the farmers.
Campus ki podam untundi. Mirror anta.
As a part of
green block lo le.
I request to host mute all the other participants. Yes, Vasudha.
Priyanka. Ostava.
I request host to please mute the other participants. Yes, he is a frequent visitor to the radio stations and also the TV channels. He has acted as a resource person at All India radio stations at Kadapa, Tirupati, as well as Vijayawada. Many phone live-in programs in horticultural crops, as well as many interviews in mango crop, which were broadcasted regularly by the All India radio stations as well as the TV channels. And he is a life member of Plant Protection Association of India, a Society of Biocontrol Advancement, and Association for Advancement of Pest Management in Horticultural Ecosystems, as well as Entomology Society of India. With this brief introduction, I invite the today's speaker, Dr. D. Srinivas Reddy Garu. So we are fortunate to have you here, sir.
Good evening, madam.
Yes, sir.
Yes, sir. I'm audible, madam.
Yes, sir.
Yeah, madam, the screen is visible, madam.
Yes, sir. You keep it in slide mode.
Yes, madam. It's in slide mode, madam.
No, sir. It's normal mode. Make it a slideshow.
I will share it. No, madam. No.
No, sir. You click on slideshow, sir. That's enough. Maximize the screen.
No, madam. It's okay.
No, sir. On your system, you maximize the screen and keep the slideshow.
That is there.
Yes, sir.
Okay, madam.
No, sir.
Second.
Means the side slides are also visible.
Srinivas Reddy,
Sir.
On your screen.
call Gopi, he will help you.
Gopi late, sir.
Leda.
Late.
But the only thing, full screen రావడం లేదు Srinivas Reddy.
Sir. Just 1 second, sir.
Okay, try.
Now it's okay, madam.
No.
Yes, sir. Now it's okay.
Yeah, now okay.
Okay. Oh,
Thank you,
madam.
sir. I welcome you, sir, to the session.
Okay, thank you. First of all, at this August gathering, I want to thank the organizers, especially the Associate Dean Madam, Swarajya Lakshmi Madam Garu, Jay Brother Madam Garu for it's not a brief, it's an extensive introduction, I feel. So, and all the course directors or journal heads, Sir Venkatramana Sir, good evening, everyone. I may not go into the details of each and every pest because maybe most of they are final year students knowing the brief pest of citrus, but only what I want to highlight is what the actually work we are carrying out at Tirupati, especially on the citrus pest management, and in context to the what the work going out at the international level. So maybe at brief. So slides are moving, madam. Okay, madam. So if you look at the citrus as a whole, wheat, baby, acid lime, and sweet orange, from the day we plant, raise the crop until we harvest the fruit, there are different complex of pests. Here we can see from citrus butterfly to the citrus fruit fly. So maybe some 15 to 20 are the major pests we could find across the country. Though sweet orange is cultivated mainly in Andhra Pradesh, Telangana, and Maharashtra, but compared to the mandarins, where in North India and Northeastern India it is cultivated, nevertheless the pests are same. So before I go into the details, nowadays even the government of India and the government of Andhra Pradesh, now everyone are pushing move towards either the research aspect or the extension aspect or even the schemes. Everyone are moving towards organic farming. So here I put a brief difference between the organic farming and conventional farming. But when we look at especially at the crop protection aspects, there is a clear-cut difference. What are the products allowed in organic farming and conventional farming? So mainly we can say the plant-based products and also biopesticides comes under organic farming. There are conventional chemical, because as we move down the slide, we could see many chemicals, or we can say the products which are allowed under organic. So this is the recently approved organic products list by the government of India under the Ministry of Commerce 2005. Here if you see, because in most of our research activity we are using light traps, sticky traps, and even the mineral oils, which has now become a significant say a treatment in the most of the pest management related to the citrus or the acid lime. So just to have our audience awareness or the students awareness, what are the permitted products under organic, what are the restricted products under organic, and what are the prohibited materials. So coming to the brief, when you take citrus as a whole, or a wheat, maybe a cyan, orange, or acid lime, or any citrus group, maybe pummel or any, see, leaf miner is a common pest. If you see the biology, the adult moths will stay or live for 15 to 55 days. Even the caterpillar, where it mines inside the leaf, lives for 5 to 30 days. So overall, one female adult, if you say, it will take about 46 to 55 eggs. So what is the typical damage? Because this is, I think, a citrus graduate readiness program being a focusing on the students. So what I thought is, let's the picture, if they are clear cut, the only typical symptom what we can see here is serpentine mines, which are silver in color. The silver is colored due to entrapped air. That's one thing. And at early stages, if you see the leaf miner, there will be curling. And if they receive a damage, it will be a leaf fall. The important damage symptom we have to remember is, whenever there is attack of leaf miner, here you can see it has always closely linked up with the citrus canker. That's one disease, bacterial disease, and there is mealybug. Because the mines, they are always on the ventral side of the leaf, which we can see where the bacteria has easily accessible to this leaf. And what we studied is, over a period of years, this leaf miner, even at the research center or throughout the farmer's field, it is present throughout the year. It has no specific period, but except severe cold and hot summer, that is April, May, I can say, for hot summer, and cold maybe December. During this period, except all the 9 months, we can see the infestation in either in the nursery or the main fields. So earlier, when we look at the general management practice, they used to recommend pruning or even neem-based kernel extract, or when you use the conventional like monocrotophos, phenolphos. That's all. I think that is in the books, it's fine. But what as ACRIP center at Tirupati, what we are doing is, we want to reduce the number of sprays. We want to increase the botanical awareness. We want to reduce the pest load or pest resistance. So with this intention, when we taken up the trial at all India level, and Tirupati also one of the main center. So we have developed one management practice, especially for leaf miner, be it in nursery or the young orchards, where initially when we spray Ajar Direct in 10,000 ppm at the rate 5 ml per liter, then after 7 days, when we spray spinosad, it is very effective against citrus leaf miner. This is one recent ACRIP technology where we could give the technology to the KVKs, and it has already used by the growers. What is the one major importance is, why we have selected initial spray as botanicals is, when we spray neem-like compounds, it acts as a repellent, oviposition deterrent, even antifodent. So after 7 days, because what we came to know is through research is, the botanicals will act mainly effective only for 7 days. So after a 1 week gap, you can spray the any chemical, which with one spray, you can easily control the leaf miner. When we take the farmers' feedback, as we during surveys, many of the farmers still they're recommending profenophos or using twice to thrice profenophos, which is again a gyrating problematic in resistance. So this is the ACRIP technology with botanical intervention, we could come out. So one more other major pest is citrus butterfly. This we all well know, we use from UG days, we study this. There are 2 or 3 species mainly, one is Papilio demolius, another Papilio polytus. So this, when we look at the larvas, they look like birds' excreta, they lay individual eggs, and the pupation also, as the larva passes the stage, it will turn from the green, and we can see the beautiful butterflies. So the student should remember, many of the pests, they are moths, but this is one butterfly which is acting as a pest. So this is the one damage when we, especially at the seedling stage, if we allow initially 1 to 2 years without any plant protection measures, there will be total defoliation by these caterpillars. So as I said earlier in the slides, that early instars, only first and second instar will be birds excreta, third, fourth, fifth will turn to the green in color. So regarding the management, these are the general above management practices like profenophos or other things. But what we could do in at Tirupati, even my colleague Dr. Shardaji also is there. What they worked is, among all the pesticides and biopesticides, BT found to be very, very effective. So here, though earlier we could see even carbaryl was effective, but to the knowledge of the audience, already carbaryl is banned. So what the alternative is, one of the best compound if you have any for citrus butterfly, that will be Bacillus thuringiensis at the rate 1 ml or 1 gram. We found this is very effective and it is on par with the chemical. But when we start using it or when we start recommending to the farmers, what is the major problem we are facing is the time of kill. Why this? In today's Indian pesticide market, only biopesticides have 2% share. 98% are synthetic pesticides. So we want to increase the usage of biopesticides. So the major problem we all well know as agriculture group or agriculture family, the time taken by these biopesticides, BT or Biberia or any biopesticide, time taken to kill is 3 to 4 days. So as a part of the research, we want to reduce the time. So thinking that view, what we done at Tirupati is, as we have caterpillars incidents both in the greenhouse and also in the field, we mixed with azadirachtin 10,000 ppm at the rate 2 to 3 ml. So surprisingly, when we sprayed October to December, during these months, on citrus, we could find the kill of the larvae. You can see the symptom. There is the upside down, the trait of disease or something. So we call. So this we could see that they are on par with Profenophos dimethoate. So what made this? The time taken generally for if you use alone BT, it is taking 4 days. When we use neem, it is taking 24 hours spray. So there is a synergism of BT plus azadirachtin. So with this concept, we for the first time have proposed a concept called BBC concept, botanical biopesticide combination. This we published recently in international paper, the Springer publication, where they accepted the concept, and we are want to start further work on these aspects where biopesticide will be mixing with botanical. So any student in the future who wants to do MSc, this is the one aspect we can look at where we can reduce the time of the kill and also the compatibility issues. So another, I request students, I think there is a Chris talk. Please mute yourself just for half an hour. Please spare me. So one more major pest what we could see is Scilla. So everyone knows it is a Diphorna citri. It is a sucking pest, hemipteran group. So what we could see is an adult Scilla lays up to 800 eggs in a lifetime, and the nymphs, the 5 nymphal stages, will complete around 3 to 4 days their life, the total development, and from the nymphs to the adults it will take 10 to 15 days. So how can we observe this Scilla in the field? Whenever you observe Scilla in the field, it will be always standing on the leaf to the 45 degrees angle. Here also we can see, that's one typical observation we can have. When you look at the damage, when you can see here, the nymphs, especially the nymphs from their bodies, they excrete the white crystals. This is the one typical symptom where easily a farmer or a layman can identify. So here you could see on a particular shoot, sweet orange shoot, before they open, before they unfold the leaves, we can see the many number of psyllids. This is the stage where they prefer to lay the eggs. That we should remember. And these are the symptoms nowadays we are commonly observing across Andhra Pradesh or Telangana or Bidar units. If you see left side, we can see the white crystals on the leaf in a large quantity, and another side we can see the death of the young shoot. So this in local language, many of the farmers from Nalgonda or even from Kadapa, Anantapur, Kurnool, they are calling Chakra Vachinsa, Chakra Tegulani in Telugu vernacular language. But this is the one Scilla damage where we can see seeing the heavy incidents occurring. Though it has been associated that citrus has been cultivated in India, but over a period of years, for the past 3 to 5 years, we are seeing the increase in the incidents. So this we can see lot number of psyllids we have found in Kadapa and Anantapur region, and this is one disease they carry, that is called greening. How do you identify greening in the fields? If you have a typical island-like symptoms here, you can see this is oftenly transmitted by the citrus Scilla, by the adults. So when we look at the survey data, or when we went for survey for the past 4 years, as I joined 17, 18, 19, 20, there has been continuously outbreak in the fields. Maybe because of the faulty practices adopted by the farmers or the not following the right recommended practices. These two communication gap has increased the Scilla incidence in the field. Other than sweet orange, nowadays in Nellore, especially from Nellore region and even Tamil Nadu, many of the farmers are calling, we are having the Scilla incidence in acid lime. Earlier, it was not much reported on acid lime, but nowadays we are seeing frequent queries coming to our farm that we are having the Scilla incidence in acid lime. So this is a cause of worry. So once it gets infestation increases to the acid lime, so the management may be taken a correct way. So when we went even for the nurseries, what is the status in nurseries? So again, we can see the Scylla. Now most of the nurseries, because of the lack of the awareness, they are supplying the same buddlings to the farmers. So we could see that already the disease load, the greening disease load is being transmitted to the field in a larger way. And as we all know in the literature or when we studied, what are the alternate hosts for citrus Scylla? One is curry leaf. So when we went to the field across our Rayalaseema region or coastal region or Telangana region, we could see now the curry leaf is attracting more of Scyllas. Maybe because of more, the crop has been brought under cultivation, or maybe some other varieties being introduced by the farmers for curry leaf cultivation, especially Tadapatri area and some other areas. So when there is no new flesh growth on sweet orange or acid lime, these Scyllas are maintained on the curry leaf. They are living on the curry leaf in the white damage. So here you can see the across. If you see we can say the long look also, we can see most of the curry leaf is acting as a reservoir host in our region for Scylla. And one more offer nowadays, everyone, we have landscape beautification, we have park being made beautiful in most of the states. So one ornamental plant, what the municipal corporation without having awareness is they are planting this orange jasmine. So we inquired with the municipal corporation of different districts. So this is one particular host which is again acting as an alternate host for Scylla. So we should be very careful when we took this as ornamental plant for planting in the parks, amusement parks, or nearby roads. So especially with the citrus Scylla, why I am more focusing on this pest is because it is creating major problem. Earlier in USA it was not there. Now after 95 it has become a major havoc. In India also, almost all the citrus group it is creating a problem. So many of the farmers, what they should do is they should have a electric traps. The main thing is monitoring. Either you go for electric trap monitoring, there you can detect only adults. But if you want to have visual monitoring, better to have a tapping method. There just you can take around 1 feet pole or pipe, just tap for 3 or 4 times. You can see many Scylla dropping on your plain paper. So that's one tap sampling they do. This is one very important. Monitoring keeps a major role in monitoring the Scylla management. So when we look at the traditional practices or the management practices, we all know removal of the collateral host baby, curry leaf for the orange jasmine, or the one promising biocontrol agent, what even we heard from NBR is Tamarixia radiata. So this is one promising bioagent where we are now have got a voluntary center of biocontrol at Tirupati center. We want to multiply this parasitoid and release in a larger numbers, because nowadays even because of organic farming, the biocontrol has taken a bright shape and many of the farmers are demanding for the biocontrol agents. So that's one aspect where any student, PhD student worked on citrus Scylla, he can choose this biocontrol agent for management aspect. And one thing, the Nagpur center, which is the nodal center for citrus, they also extensively worked on this biocontrol agent called Melada bonensis, and they released extensively and they could get a good reasonable control. So here you can look at these photographs. The above 3 photographs shows the promising parasitoid called Tamarixia radiata, which is already available in Bangalore. They are also ready to give to any farmers who want to release it. And you can see from the nymphs, it is an parasitoid, where it attacks the nymphal parasitoid we call. And the down one, you can see the Melada bolensis. So these two are the very promising
Sir.
Sorry, there was a power breakup. Is it visible, madam?
Yes, sir.
I'll go to the slide.
Yes, sir. Visible, sir.
Sorry, there was a power breakup. I am extremely sorry. So what we could do it, these are the general earlier recommended practices for the SILA, like imidacloprid, dimethyl, arconalpha, that everyone knows. But what we could do recently is whether is there any specific chemical which can effectively control citrus we studied at ACRIP. We found foliar application of novaluron, especially 0.5 ml or azadirachtin petroleum spray oil found to be very effective against SILA. But recently, what is the work going on at international level, especially on the citrus psyllid? Maybe reducing pesticides. So one very good work which is being carried at Taiwan and Vietnam is psyllid repellent research. So especially they have taken the guava plants. When you intercrop guava with the citrus, you have a lot of reduction of psylla infestation, and they found out what is the one particular chemical which is responsible is dimethyl disulfide. So this is the one aspect where we can say it is at the infancy stage in Indian context, where no one has attempted to study this using of intercrop of guava in citrus to reduce the psylla damage. So though we proposed recently, we have come up with an submitted a project to the NASF that is called as RAT, Repellent Application Technology, where we want to use guava in the citrus for management of psylla. So here you can see on the photograph where they are intercropping the guava with the citrus, there is a lot of reduction in the psylla. So in the advanced work, as I told, they are extracting that dimethyl disulfide purity of the in the laboratory, and they are spraying to the citrus sweet or other citrus groups so that they are getting the repellent action. So that's one particular aspect research going on at citrus psyllid in other countries. What is the one recent work being carried out at USA level is insectigation. So as many of the students know, they know fertigation, the application of fertilizer. Now there is one new concept emerging. Maybe in the Western countries already they are practicing long back, the insectigation, that is application of insecticide through drip. So this is the one new emerging, what you can say, researchable issue in India, where the countries already, like University of Florida, where they are doing a lot of extensive work on psylla management. So application of insecticide through drip. So especially like a neonicotinoid compound, be it maybe imidacloprid, thiamethoxam, or clothriane, they are using extensively for insectigation. Even recently, we have come up with a review article and submitted to the journal insectigation as a new approach to pesticides. So the other one particular, if you see at there is one interesting finding recently in 2018, when they applied thiamethoxam through drip at 4 grams and 10 grams, this is the new work, that's why I highlighted, they are getting control up to 60 to 90 days. And they found that even if applying the insecticide through drip or soil application, they are not finding any residues. So this is a researchable issue in India, especially in USA. They are recommending that before the orchard starts bearing, maybe 4 to 5 years, many of the insecticides, especially the neonicotinoid group, they are applying through soil and getting effective control. So another, if you look at the recent, one more interesting work, what they are doing is in USA, use of organic compounds. So they like this organic 1 module, organic 2, organic 3, and conventional. These 4 modules they have taken in 2014 and 15. They have sprayed on Valencia variety, sweet orange, and they found that what you can say biocontrol agents are found in large numbers and good numbers, especially in the organic 1, 2, 3. If you see at the model, like azadirachtin, there is a one biocontrol, which is a organic module, pyrothrins, mineral oil. When you look at the larger conventional. So especially when you look at the data, what they got is in most of the organic, you can see the good number of natural enemies surviving. They released these natural enemies and found a very effective control of this Citrusilla. So this is one researchable issue also in India, where we can compare organic with the conventional in the management of the Citrusilla. So the again recent one is the use of protective covers. They put it in a simple form called cups, where each plant is covered with a protective screen to avoid the psyllid, maybe for 4 to 5 years, and they are seeing the efficacy. And the research says they are able to control till 80% without using any insecticides or minimal sprays. This is a one new research aspect where they are taken up in a larger scale in Western countries. So this is also to think of in our MSc or PhD program, any student is interested. So one more important sucking pest we are seeing is citrus aphids. So you can say brown and black aphid. So the congenial weather means one thing we should remember: this cool weather favors the aphids outbreak, and they can complete in life cycle in 1 week. And one adult aphid may lays up to directly because of parthenogenesis 4,400 nymphs, and 25 to 30 generations they can complete in 1 year. So this is a very, you can say, the population build up. That's why this is in a short period. So this is the citrus aphids. You can see on sweet orange in a large numbers gathering, congregating at the leaf tip, sucking the sap, and they transmit one important disease called Tristigia. This citrus aphids. Though nowadays in the field we are not finding this disease, but aphids you can see in a large numbers in the fields. So earlier, many conventional management like phenolphos, dimethoate, conventional OP, they were recommending imidacloprid. But recently, we have come with ACRIP recommended practice from Tirupati Center. That is, initially when you spray, again azadirachtin 10,000 ppm, 5 ml per liter. With 1 week interval, again you are spraying thiamethoxam 0.25 grams. We can check the population of its effectively. So one more important sucking pest is citrus black flies. Though the incidence is very minimal, even in the farmers field and even at our Tirupati Center, but we could see somewhere because of the faulty practice or using of more pyrethroids, this pest is becoming nowadays. So the same management can be adopted. And one more important pest is mealybugs. So everyone knows the mealybugs are very difficult to control. There is a Planococcus citra is the common citrus mealybug. Because of the waxy covering, it's very difficult to control. Though in the fields, we are not finding any difficult. But what difficult we are finding is in the screen house. So citrus cultivated under protective screening will definitely have a havoc with this pest called mealybug. So when we gather the data or the species, so we could find a other species called spherical mealybug. Nepococcus viridis is not Planococcus citrae. So this, if you can see, they congregate. This is the ovisac it is called, where they lay large number of eggs, because as we know, mealybugs doesn't go natural reproduction sexual. It is goes under parthenogenesis. So it will lay eggs in nymphs in large numbers about to hatch. So initially, if you see this, why it is very difficult to control. Initially, it is very easy to control because there is no wax covering. If you see the photographs, you can see the all light pinkish or red color nymphs in large congregating in large numbers. So at that time only it is very easy to manage. Be it may be biocontrol or it may be pesticide. Once you have a second and third instar, you have a mealy coating. Here you can see many of the mealybugs secrete their wax. Early stages that doesn't have the wax glands, but when they reach the later stages, we can see the wax gland secreting mealy, and you can lot of mealy coating. This one, the aspect which is creating problem in management. So for the benefit of the audience, this citrus mealybug, you can see transmits a disease called citrus mosaic. So, and this is the down is citrus greening. You can see the island symptoms, citrus mosaic symptoms, and the deficiency. Often when we go to any citrus crop, be it maybe any species, we often confusion which is whether it is virus, whether it is bacteria, or deficiency. So as an extension agent, as an extension personnel, we should be clear-cut able to differentiate whether the particular problem or in a correct diagnosis, whether it is a virus or a bacteria or nutrient deficiency. So we all know that we can use yellow chiquitras for monitoring. So on aphids, so what the ACRIP objective was, whether we can reduce the number of sprays, whether with single spray we can manage all the sucking pest complex in sweet orange or citrus. So with that intention, across around 8 to 9 centers in citrus at acre fruits, we could come out with one technology called, when we spray azadiractin 10,000 ppm at the rate 5 ml, followed by a thiamethoxam at 0.25 grams, we can manage aphids, black fly and psyllids, because all these three pests usually generally occur in winter months, especially on the young foliage. So we all know that as a part of ISRHU and also the other staff, we are raising the mother plants in the screen house. Now many of the farmers are raising the screen house, the sweet orange mother block in the shade net, which is not advisable. So better to have a screen house when you want to establish a nursery. So here, what was the need to establish a screen house to raise the sweet orange mother? Because as we know that these two are important vectors for the diseases. So if you see the whole size, the micron size, what we use for the screen house depends on the pest. So especially for the Scilla, we are using 40 to 50 micron, or the aphids, we can see the pest, especially when you leave, the least is thrips. If you want to avoid the thrips entering the screen house, it is highly not, you can say impossible, because almost we can't have that much thickness of the mesh. So these are the, depending on the need, be it maybe any crop, depending on the need, what pest you want to exclude from the screen house, we have to choose the micron size for building up the screen house. So these are the other weevils. There are 4 types of weevils in sweet orange, where generally they do defoliators, but not much significant damage. With general sprays we can manage. But these are the 4 species. They sometimes they look similar also, but these are the 4 different species we observed in sweet orange. And one more, even from ages we could see the pest is snow scales. We could see earlier it was mainly we could see on acid lime, but nowadays we could see also on sweet orange. So if you see the white patch on the main trunk, we could see there, like how we paste the lime paste. Similarly, we can see the snow scales damage on the main trunk. And here I put one photograph, because these scales, why it is very difficult to manage, because the scales sit one above the other, maybe some 8 to 10 layers. How these people have fallen one above the other. Similarly, way on the tree trunk, they sit one above the other. Some 7 to 8 layers we are able to find on the field. That's why it's very difficult to manage. And if there is no management practice adopted, no intervene, then we could see the bar split. So many of the farmers, or we can see in the field, when there is a snow scales damage on the trunk, we could see the splitting of the bar. This one, severe damage of snow scales. And often we say that the snow scale damage is coinciding with the peak summer season. But nowadays, due to change in the weather conditions, even we are seeing in the winter months. So this is the one life cycle. Here, one thing very important is, initially up to first mold, both will be same. You can't differentiate which is male, which is female. When the first instant undergoes mold, there will be 2 sexes. One will be female, another will be male, where it will go to the different stages, pupa, pre-pupa, and they fly. They doesn't stay on the trunk. But mainly the females will have a, what you can say, which is reproduces and stay in the trunk and damages the crop. So when we have the close-up photographs, these are say hard scales, soft scale. There are many different scales, but snow scale is a hard scale. This is the male, you can see, and this is the female. You can see how it will lay the eggs in masses with again waxy coating, because all we know, mealybugs and scales, they belong to the same family, Pseudococcidae. So one particular aspect, many farmers are querying us whether we have any effective management for snow scales on acid lime. So we taken one particular researchable issue on this as a non-plan. So we have taken variety biology and done some work. Initially, the old practice was they rub the main trunk with the gunny baits. That all we should do. We are doing it, and the old recommendation for dimethyl imidacloprid. But initially, what we done is, if we mix any particular oil with the insecticide, whether the efficacy of the pest is more effective or less effective. If it is effective, how many days it is effective? So with this researchable issue, what we could see that here, up to 7 weeks after spraying, we could see the effective control of snow scales, especially when you mix insecticide with petroleum spray oils. And in the coming slides also, I will tell you why these oils have become more important in management of sucking pests and also in the citrus. As compared to the dimethyl, we could see that around 68% reduction over control compared to the individual insecticides. So now many of the students who are also maybe doing post-graduation or at the end of the completion of their degree, or to the benefit of audience, nowadays there are many are asking for mineral oils use. So we can also call AMOs, HMOs. I don't want to go into the details. They are also called white oils. They are quite popular in USA, where they are approved in organic also, in India also. So one important thing we could see is the mineral oils, how safe they are for use in the crop. It all depends on one particular parameter that is called unsulfonated residue. So general, you should remember is, as a general rule, higher the unsulfonated residue in the mineral oil, better is the safety to the crop. That one thing you should remember when you use mineral oil. So there are the carbon ranges from 21 to 23. So what are the different pests where we can use this mineral oil as important management component? We can see scales, which is becoming a major problem, where we can mineral oil has a good amount of control. Citrusilla, mites, mealybugs, thrips, mosquito bugs, even some discs also they are using. So how do these oils act? They are mainly as asphyxiation. They block those spiracles and kills the insect. So especially in citrus, what is the dosage we can use? If you use for mineral oil, that's one aspect also we are doing researchable issue. If you use 0.5 ml, less than 0.5%, we are getting very effective citrus leaf miner. Or if you want to use for schools, we should use minimum 1%, that is 10 ml per liter. So this is again recommendation for citrus, where during cooler periods we use for control of citrus leaf miner. So only thing is this, we should not, what you can say, at higher temperature, this mineral oil should not be used, and they should not be mixed with copper compounds. So these are some commercial mineral oils which are available in a market like paraffin coil, which we are using for management. So one more very important pest after fruit formation is mites. Everyone knows water mites. Both nymphs and adults suck the sap from the leaves, fruits, everything. But there are two important species of mites. One is green mite in sweet orange or citrus, and there is rust mite. So this rust mite, we can say in local language it is called mangu because of the dark brown skin, and in acid lime also we can type this type of symptoms. Now, even when we grow across different regions growing citrus, why this citrus green mite is becoming a major problem? Earlier it was there, but throughout the year we are now observing this. We could see any alternate host. So based on that, we started doing research and also collecting the samples from the field. One, the green mite, the one important alternate what we could see is curry leaf, and another is neem. So when there is a no fruit, when there is a particular leaf, when the farmer is spraying the acaricides to control the green mites, they are retained on the, because as we know, many of the farmers, Indian farmers, they grow neem on the bunds. So especially this is acting as a reservoir or alternate host for green mites. These are the different symptoms. Many of the farmers are getting confused with some nutrient deficiency. So these white patches we can see, these are each one colony. They are green mite colonies. So the literature says, or what we could see, there is two species. One is Schizotetranicus hindustanicus and Baltazari, but still a lot of clarification is required which region has this species. So if you would see from our microscope what the photographs we got, you can see here the green mite eggs. They lay individually along the midrib only. They lay the eggs on the citrus, be it maybe acid lime or sweet orange. That we could observe. And one typical is after the female mite lays eggs, it forms a webbing as a protective cover on the egg, and it protects its young ones. So even once the nymphs hatches, they also remain under webbing, suck the sap, and after maturity, they come out. Here you can see a lot of the mites activity under the web. So again, we could find whether neem, those symptoms we observed, whether it is retaining the mites. So we could see when we collected the neems from different fields, we could see these are here also a lot of mites activity we could record in the microscope. And surprisingly, this neem nowadays becoming as a reservoir host both for rust mite and green mite. Here you can see the individual eggs are laid of green mite, and you can say the spherical shells, there are rust mites. So this is the photograph we took from the neem during the summer months. So many of them know there are cryptic sibling species in insects, where through morphology we can't identify species. Though we identify, but the accuracy remains a question. So with this keeping in mind, we started working on molecular work, because nowadays the using of DNA, as maybe someone from the audience also may be working on it. So we are using this DNA barcodes using specific primers for identification of green mites. So we could see, we collect the mites from these alternate hosts or also the main hosts, and we could get the band, and we are sent for sequencing, DNA sequencing. And again, citrus green mite. So these are the again different mites. Maybe Eutetranincus also we could see some in citrus. So in general, everyone knows that we should use acaricides for management of the mites. The old conventional were dicophol, wettable sulfur. No more, they seem to be effective at the field level. So keeping that in view, at the ACRIP workshop regularly, they ask for management of mites, be it maybe green mite or rust mite with new acaricides. So among that, we find ethion 50 EC at 2 ml or propargite 1 ml found to be very effective. Though it has been done the work 15, but recently, any technology, if you want to recommend, we should have a residues, because these are all pesticides. So recently, what we have also done some residue studies, whether the acaricides what we are recommending at the field level, whether they are safe or harmful. So we could see most of them, they are safe for kill of the mites. So recently, last year only, we have concluded this technology, that is the new acaricide what we are recommending for, be it maybe green mite or rust mite or Eutetranincus, whatever species you find in the field, citrus field, we could recommend spiromescin at 0.39, found very effective with 20% increase in the field. But one particular aspect, what was lacking is many of the farmers, even before just 1 or 2 months of the harvest, they are asking, sir, if you spray acaricides, whether we can control mites. So one particular aspect, what we have observed is keenly is the application period is very, very important for management of the mites. As all we know, in sweet orange, like acid lime, it takes 5 months from flowering to fruiting. Like in a case of sweet orange, it takes 8 months. So during this period, 9 months, every month we can't spray the acaricide. So keeping that in view, whenever we recommend any acaricides for sweet orange, the application period is very crucial. So we should apply before 2 months. That too, first spray maybe using ethion or propargite, and last spray before 2 months. Means before the fruit reaches the 4 centimeter diameter only, we have to complete the spraying of the acaricides. Once the fruit stage crosses 2 months, doesn't make sense for application of acaricides. That's what we have come out with the conclusion with the acaricide management. One more often we see nowadays because of the black thrips. This even recently we got 2 days back from Anantapur. This black thrips been reported from citrus. We doesn't know, but we are yet to move for the survey. But the general thrips, what we could see is that is Cytothrips setae. The one thing the students should remember is many of the stages we could see above ground. But one important way why we are not able to manage or control these thrips is because the 2 stages are in soil. One is pre-pupa, another is pupa. They are remain in the soil. Irrespective of the species, majority of the species undergo pupation in soil. So only the first instar or second instar are causing significant damage to citrus. So and that too we know as the temperature increases from February onwards till June, we could see the thrips significant damage in the field. These are the different species we could see in citrus. And how do you identify the thrips damage on sweet orange? You can see the curling or cupping, what you can say, or along the midrib, 2 white wavy lines. If you could see on sweet orange, that is a thrips damage. So here also we can clearly see 2 wavy lines running across along the midrib, that is attacked by thrips or general upward curling. But always in the field, you doesn't find the upward curling. This we could even see the damage in screen house. And in fruits, when we say irrespective of the stage of the fruit size, it is damaging. So we can see that they scrape the epidermis, they suck the sap, they reduce the fruit size, and you can see the, what you can say, ugly look. The cosmetic appearance is damaged because of thrips. And you could see that they complete the generations in a very faster way, maybe 8 to 10 when the weather conditions are congenial. And one important thing is here, I put a arrow mark, they remain in the sepals of the young fruit. Once the fruit formation is starting, they remain in the sepals. That's why whatever the pesticide or any botanical or biopesticide you are spraying, it is not able to reach the thrips. That's very difficult even with the chili or the other fruit crops. Yeah, and one typical symptom, what we can see when the fruit is maybe some mid stage of the fruit, we can say a ring white symptom, white ring like symptom, you could see that is thrips damage. Or we can say mottled patches on the fruit, that is also thrips damage. So general, earlier we should say plant sticky traps depending on the area, yellow or blue both can be used, or removal of weed hose, especially Hypomea batata or other weed hose, or earlier the recommendation was dimethoate or asphate. But now again, as I told, with the botanical intervention, we could come out that azadirachtin 5 ml initially when you spray after the petal formation. Here I have given after the fetal formation, you can say the stage 4 is very crucial for thrips management. At this stage, you spray azadirachtin botanical, and after 1 week, if you spray the spinosad, which is organically approved and safe compound for natural means or other human consumption, so we can very easily control thrips. Again, after 2 months, if you are spraying for thrips management, by the time you could see what are the below photographs when the damage has been occurred. And one more. When the fruit starts maturity, just 2 months before maturity, we could see a citrus fruit sucking more damage. So what are the host crops? Guava, pomegranate, citrus, mango, papaya. And recently we have come across even papaya is causing significant damage to papaya also, and even tomato, everyone knows that. And they are nocturnal feeders. And they are cross, wherever citrus is cultivated, they are there. And what are the different species? So by looking at the beautiful moths, we can say one is Eudosima materna, Phallonica, Janata, that is Castor semiluper, and Homiana. These are the 4 species commonly found in our area damaging citrus. So we all know that generally in Lepidopterans, this is for the students' information, the caterpillar do the damage, but in citrus fruit sucking, but the moths do the damage. So the caterpillars, the female moths, mainly oviposited on the weed hose, belong to the Mealybugs family. One is Tinospora cardifolia, another is Cocculus hirsutus, and Cocculus pendulus. These are the 3 weed hosts where the caterpillar feed on these weed hosts and complete the life cycle. So there was one recent study where they took 12 hosts and say that where the larval could complete the mortality life development. So they could say that on Tirupati cardifolia, in Telugu it is called Tirupati, it could successfully complete its development and enter into the pupal stage. So how does it damage? Everyone knows that adults penetrate the proboscis in the rind of the fruits and suck the juice. Once the particular portion, it sucks the juice. From there, you can see the fermentation smell is coming out, and it is prone to the other secondary pest attack. And the last stage is the dropping of the fruits. Significant damage we can see, irrespective of course, maybe pomegranate or even citrus, we can see up to 40% to 50% damage. So though we are writing or recommending the students or recommending, but what are all the management I have written here by putting even the solar light trap, removal of feed hose or poison bait, or none seems to be very effective against fruit sucking moth. That is a proven at the field level. So few research work what in the weed hose where the caterpillars are present. So BT found to be 2 grams per liter or Pseudomonas luminescens vivaria found to be very effective to kill these caterpillars on the weed hose. So, and as we know nowadays, everyone are coming with whether we can use solar insect trap for trapping all the insects in citrus. So recently, the net cap of the government of Andhra Pradesh has told us to evaluate to YSRHU to evaluate the solar insect traps. So these are the specifications we established in the field. These are the different insects we could collect. But the preliminary observation, what we could do is the solar insect traps are doing more harm than good. You could see most of the dragonflies are getting trapped. Most of the scavenger beetles are getting trapped. Most of the dung rollers. Means in ecosystem, these insects are required to maintain the ecosystem. So if these insects are getting trapped, definitely it's doing more harm than good. Only thing is, we could see good number significant of white grubs, which is a major pest in groundnut and sugarcane. So in those crops, maybe we can think, but not in as of for horticulture crops, because nowadays many farmers are asking solar insect trap petachasar emana pani chestundan. So preliminary observation we have 1 more year to go for further to come to a conclusion. So when we discuss with the ACRIP, how we can control fruit sucking moth, because it is a nocturnal feeder, it comes at around 7 o'clock and moves before 4 or 5 o'clock. So whatever insecticide you spray, it not effective. And the IPM practice also, they remain only 20 to 30% effective. So keeping that in view, in the ACRIP GD 2015-16, they have come out with the Nagpur, the nodal center has come out with the repellent oils. So whatever all the listed oils here, they're all repellents and they are organic based. So these oils were tested at Tirupati Center for 4 to 5 years. And among all the oils, we could see that the neem oil and the petroleum spray oil, neem oil maybe 10 ml per liter petroleum, they are found to be very effective against food sucking moth when you spray at color breaking stage. Even yield also significant higher than control. So this is the one, maybe in the coming GD we can conclude where we can recommend the oils, either maybe citrus, pomegranate or guava or banana or tomato for management of fruit sucking moth. So we could, this is the stage, the first stage where the fruit sucking moth attacks. That is called the color breaking stage, just 2 months before harvesting. So the stage is very important for application of the repellent oils. So if you apply at this stage and after 1 month, maybe neem oil intermingled with the petroleum spray oil, you find very effective citrus fruit sucking moth control. So many audience may thought, what are the new pests any reported in the citrus? Recently, during the COVID break, many of the farmers couldn't harvest the sweet orange crop. When we do the survey, they come with one citrus bug, which is not a new pest, but it seems to be a new pest on sweet orange. It's a Leptoclossus species, citron bug it is called, but not much significant damage, but it damages the fruit, and there is a damage of some around say 13 to 18%. And we could see any alternate host, it is damaging banana and also the pomegranate to a certain extent, but still we could not see the damage in 21. Maybe more years required to confirm whether it is really a species causing significant damage to citrus or not. So one more new aspect, what we are doing is reduce the number of sprays to manage all these 3 pests or 4 pests. So bagging is one aspect we are looking at. Bag the individual fruit and see whether we can, this is only a preliminary trial we have done. We got some encouraging results and we could see further fruit covers if we use, whether we can reduce the number of sprays and arrest the pests. So this is my last slide. Thank you for your patient listening. Again, I want to thank the organizers, Dean B. Srinivas sir, Swarajya Lakshmi garu, Jayaprada Madam garu, other core directors, professors, journal head, and my colleagues for patience listening. Thank you.
Thank you, sir. I request the participants, are there any doubts or clarifications you need?
I request student participants, you are having any queries, please put it in chat box or you can interact directly with the Srinivas Reddy sir. Very excellent presentation.
Yes, sir.
It's excellent session, Srinivas Reddy garu.
Thank you, madam. Thank you, madam.
Observing the crop, and nowadays it is becoming very, very important to go for organic way of controlling insect pests.
Yes,
Observing the pest life cycle, everything, each and every point is important. That are all are covered here today. Insectigation is also, it's a new work that is going on.
Yes, ma'am. Yes, ma'am.
Yes, sir.
Though in India we have not have any insecticide approved for insectigation.
Yes, sir.
But any student can take up that particular aspect and study.
Yes, sir. Yes, sir. It can be taken up by PG students.
Yes, madam.
For their PG programs.
Sir, I have a small doubt, sir.
Please, madam.
Sir, regarding the Scylla control, you told that there are some experiments going on growing guava in between the citrus plants.
Madam.
Are there any common pests between these two crops, sir?
I don't think, madam. Maybe only fruit fly.
Yes, sir. But fruit fly is a major pest in guava, no, sir?
Yes, madam. That we can manage, madam. But as a trap crop, we can plan. Not as maybe in Vietnam, they have done some studies. But as a trap crop, we can plan guava. We also planted around greenhouse, sir.
హలో శ్రీనివాస రెడ్డి గారు హలో
చెప్పండి సార్.
శ్రీనివాస రెడ్డి గారు డాక్టర్ రామయ్య.
చెప్పండి సార్.
హౌ ఆర్ యూ?
ఫైన్ సార్.
Okay, so extempore presentation. Once again, congratulations to you.
Thank you,
So
sir.
one small information, whatever you told is that is correct on the sucking pest. Actually, in sucking pest, according to the literature, I heard that all the sucking pests are predominant in the high temperature prevailing conditions.
Sir.
I studied in the literature.
Sir.
But here, the aphids and also some of the mealybugs or some of the thrips, according to your voice, I heard that winter conditions are prevailing.
Yes, sir.
So I need some clarification on this one.
Sir, actually, we have finding even snow scales in the winter conditions. It's not a problem in open field condition. It is a problem only in protected conditions, greenhouse.
Okay.
Here the temperature is comparatively lesser, 3 to 4 degrees to the external conditions.
Okay.
And even Silla we could see, because this particular year 22 we could see that the cooler months have prolonged, so we could see Silla damage even in the winter months. Generally we do December, January, but February, March.
Only in greenhouse condition. You have observed this greenhouse condition.
Yes, sir. Yes, sir.
Okay. So that is nice. Okay. Very good. Nice. So actually, I heard in the literature, all sucking pests are high temperature prevailing conditions. They are the predominant. Their development is the enormous. That is the literature I heard. But your voice, I heard that is the winter months are prevailing for the thrips and also some of the aphids, some of the mealybugs like that. Okay,
sir,
that is only in the controlled condition you are telling. Okay.
Yes, sir. Citrus, Silla and aphids we can see in winter also, sir.
Okay. So
we can see winter in under screen house, but Silla and aphids we could see in winter.
Observed Silla in curry leaf in summer months also, but according to your observation, that is winter also the predominant.
Winter also, yes, sir. Yes, sir. Yes.
Okay. So and also one more thing, the summer indicate the season. There is no, again, and we tell the summer season, the winter season like that. Winter indicates the season. That is winter or summer. There is no adding of the season because that is grammatically. Because you are the extempore in presentation. So the summer is enough or winter is enough. Okay, right.
Thank you, sir.
Okay, congratulations. Congratulations very much. Thank you.
Ramaya sir, we all know that you are following the session along with the traffic, sir. You are controlling the traffic.
Thank you, Jay Prada garu. Thank you, Jay Prada garu.
Thank you, sir. Thank you, sir.
Okay, right.
Sir, Srinivas sir, I have one more doubt. Regarding the mealybugs control, you means not only mealybugs, for different pest control, you recommended the screen house with different gauge, sir.
Madam.
Screen house.
Yes, madam. Yes,
sir. You recommended the first one 36 microns, sir. Is there any problem if we grow them in 36 microns? We can control all the other pests also, no, sir?
No, madam. Only particular pest. Suppose we have now at Tirupati, we have screen house only for Silla. But we could see inside the screen house, we could see the aphids entering into the screen house, the thrips entering into the screen house, the mites entering into the screen house. So it depends on the what species you want to exclude, because we have in citrus the greening, disease-free greening buddings we have to supply to the farmers. So Silla is a major problem. That's why they have chosen the screen house with a particular mesh which can exclude Silla but not other pests.
Depending on the location where the pest is.
And, what pest you are targeting? If you are targeting thrips, definitely we have to go for 176 micron. Thank you.
Thank you, sir.
Thank you, madam.
Are there any other clarifications needed for the students? Any other participants, the staff? Sir, in chart, there is a question that what about severity of damage in citron bug?
No, madam. Only 1 year we could observe because in COVID 2020, none of the farmers could harvest the fruits. So they left the fruits on the tree itself for maturity, ripening. There we could see damage on citrus. But last year, though we went for survey, we couldn't find any citron bug. Maybe more years required to confirm whether really it is causing a significant damage to citrus or not.
Earlier also they were under the threshold level, sir.
Threshold level.
Any other questions from the participants? So if there are no questions, sir, Reddy sir.
Yes.
The presentation and interactions are over, sir.
Okay. Are there any queries from the student side still?
Only staff have needed clarifications.
Yes. The topic is quite interesting and the presentation is also very good.
Very outstanding presentation.
Yes,
sir.
outstanding presentation. Very nice presentation. Thank you, Dr. Srinivas Reddy garu, for your excellent presentation.
Thank you, sir.
On the topic, I think your presentation has really, enriched the knowledge of our students, particularly on this citrus pests and their management. I think you have given the latest problems also identified in citrus crop. And also some of the measures to be taken to overcome the problems. So it's really quite interesting, your topic. And I once again thank you on behalf of university and the staff and students for your nice presentation.
Thank you, sir.
And for sparing your valuable time, though you are busy with your regular activity there at Citrus Research Station. And I'm once again very much thankful for your nice presentation. May I now request Dr. Jayaprada to kindly propose formal vote of thanks to today's guest speaker.
Yes, sir. Thank you, sir. Before going to the formal vote of thanks, Dr. Srinivas Reddy, sir, we are fortunate to have you in this session, sir. And I am very happy to inform you that you are one of the favorable teachers of our students, sir. When even now, even though you are not here working at the college, the students still remember you, sir, as one of their favorite teachers, sir.
Thank you, ma'am.
Sir, can I propose a formal vote of thanks, sir?
Yes, please,
Yes,
Dr. Jayaprada.
Yes, sir. Respected dignitaries gracing today's program, Dr. T. Janakiram Garu, our Honorable Vice Chancellor, Dr. YSRHU, Dr. A.S. Padmavathamma Garu, Dean of Horticulture, Dr. YSRHU, all the respected university officers, and Dr. D. Srinivas Reddy Garu, Senior Scientist Entomology, who is today's guest speaker, all the associate deans of the colleges, Dr. K.T. Venkatramana sir, Principal Scientist and Head HRS, Anantarajpeta, Dr. R. Nagaraj Garu, Principal Scientist and Head CRS Tirupati, Dr. C. Madhumati Madam Garu, Principal Scientist and Head CRS Petluru, the course coordinators, organizing committee members, faculty members of all the Dr. YSRHU, our beloved students and all other participants, a very good evening to one and all. On behalf of College of Horticulture Anantarajpeta and entire fraternity of the institute, I take this opportunity to propose a formal vote of thanks with a deep sense of gratitude to all who have helped us in organizing today's session. I take this opportunity to profusely thank respected and our most distinguished chief patron, Dr. T. Janakiram Garu, our Honorable Vice Chancellor, who is the torch bearer of this event for leading and inspiring us in working towards the goal of conducting this Citrus Graduate Readiness webinar. I'm running short of words to express my humble thanks to Dr. A.S. Padmavathamma Garu, Dean of Horticulture, for her constant guidance and moral support in all activities of our college. We are highly obliged to you, ma'am. And I thank and extend gratitude with a due respect to all our beloved university officers for being with us in the entire program. I immensely thank Dr. D. Srinivas Reddy garu for readily accepting our invitation and enriching all the participants, not only the students, with the very valuable information regarding the sucking pests, their symptoms and control measures, especially by following the organic method of control. For sparing his valuable time, for making excellent presentation and making this session very interesting and meaningful. The lecture was elaborative, very informative and useful to the young budding citriculturists, sir. We have been fortunate enough to have our beloved Associate Dean, Dr. B. Srinivas garu, very motivated and dedicated senior colleagues, Dr. K. Swarajyalakshmi Madam garu, and all the teaching and non-teaching staff. Most importantly, the students of this College of Horticulture, Anantarajpeta, who worked very hard and carried out all the works allotted very diligently. Sir, with your permission, we will end the session, sir. Thank you, all the participants.
Thank you.
Thank you, sir.
Dr. Thank you. Once again, I extend my heartfelt thanks to Dr. Srinivas Reddy garu.
Thank you, sir.
For being with us for today's program.
Sir.
And I'm also thankful to Dr. Jayaprada garu and also senior colleague Dr. Swarajyalakshmi garu, all the staff members and students who have attended today's program. Thank you once again.
Thank you, sir.
I think we can close this program.
Thank you, sir.
Thank you very much, sir. And I am
you.
happy all the participants are enriched with this knowledge and they utilize them in the future.
Yes.
Yes, ma'am. Yes.
Thank you. Have a good day.
Thank you.